We have a really very special presenter today, a scientist who contribute many different in different aspects of photoreceptors to biochemistry, and we're truly delighted. And with this very prolonged introduction, I will ask Dominic to introduce our speaker.
Good morning, everyone. My name is Dominik Lewandowski, and I am honored to introduce our esteemed speaker today, Dr. Naash, who will enlighten us with her talk titled Usherin and its Role in Health and Disease. Dr. Naash holds the prestigious position of John S. Dunn Endowed Professor of Biomedical Engineering at the University of Houston, Texas. She also serves as a professor in the Department of Biology and Biochemistry in the College of Optometry, a testament to her broad knowledge and interdisciplinary approach. Dr. Naash earned her PhD in biochemistry in 1990 from Baylor College of Medicine in Houston, Texas. She then pursued a postdoc at the Cullen Eye Institute at Baylor. She received her first faculty appointment at the University of Illinois, Chicago in 1992. Dr. Naash's groundbreaking research on genetic mutations associated with hereditary retinal and hearing disorders has earned her widespread recognition as an authority in the field. Her work on viral and non-viral ocular gene delivery has not only garnered international acclaim, but also significantly advanced our understanding of these conditions. Dr. Naash has maintained an impressive research portfolio over the last decades, and she is currently the principal investigator on several NIH-funded grants. Throughout her career, she has received prestigious awards and published many excellent papers in top journals such as Journal of Cell Biology, PNAS, and Journal of Clinical Investigation. Let's warmly welcome Dr. Naash to our distinguished seminar series.
Thank you very much. Okay. Yeah. Oh, yeah, it is on. I'm set. Good morning, everybody. Are you guys happy today?
Yeah.
Thank you, because I am so happy. Thank you so much for the kind introduction. Thank you, Krishna, for inviting me to come here and to meet all of you. Some of you I know many decades ago, along with other people on Zoom. So I wanted to acknowledge this invitation. I'm grateful for it. And also I'm grateful for Vladimir and Maya for hosting me and giving me such a welcome yesterday dinner and a brief lunch. Thank you so much. And I look forward to meet all of you this afternoon and throughout the day. So based on what Krishna asked me to present, when she sent me the invitation, she asked me to speak about Usherin. So I decided to focus 100% of my talk on Usherin, to look at the role of Usherin in health and in disease situation. So before I start, I would like to acknowledge our group, current group here, their list, and they are all here. That Moyed and I have been working with since we moved to Houston from University of Oklahoma. So today I will speak about the work of Lars Stubbe, who joined our lab about 5-6 years ago, and he came from Uwe Wolfram lab. He trained in his lab on the cilia, so it was great opportunity for us to have him, and he has been extremely productive since he joined our lab, and he is the first author on our Nature Communication work that we just published last year on Usher syndrome type 2A. I will also speak a little bit about the work with Stephanie, which is still ongoing, and Stephanie just joined our lab about a year ago. And our lab manager, Mustafa, who did majority of the work that handled the animals and a lot of characterizations of Usher models. And then also talk about some of the work that had been done by our prior graduate students who left our lab already. And I would like to acknowledge the collaboration that we have, amazing collaborator from June Young, who shared her precious antibody against the C terminus of Usherin. And you could see how this antibody became instrumental in our investigations on this topic. And Dominic Kosugrove, who we indebted to him for training us how to work on the cochlea, which is really not a simple organ to work with or to isolate. Uwe Wolfram shared numerous antibodies with us that we were able to use in our research. So let me have a few minutes to talk about Usher syndrome, if you guys are not aware of it. It is autosomal recessive form of a blindness and deafness, and it affects about 4 to 100,000 people newborn. There are 3 types of it, and the differences between the type 1, 2, and 3, it's based on the onset and the phenotype that appears in these patients beside deafness and blindness. And so far, 13 genes have been associated with Usher syndrome. 6 of them are listed in here with USH1, and 3 associated with USHR2, and only 2 genes associated with USHR3. There are other sites that are known to link to Usher syndrome, but they have not identified the associated genes. There are 2 other genes, which is like PDZ D7 and CYP250, which are thought to be also involved in Usher syndrome, but they have not been allocated to any of these subtypes. Now, USH2, which is the one that we have been interested in because it is a common one, it associated with almost 50% of all Usher syndrome cases. And currently, there are no effective therapeutic interventions. There are attempts, but none of them have been shown to be very effective. So there are a lot of challenges associated with Usher syndrome, and I wanted to bring it up here to let you know how it is very difficult to work with disease and the associated genes. The genes are very large, we all know that, and identification of the mutation is extremely difficult. And that's important for genotype-phenotype correlation and also very, very costly. That's why progress in this field has been very, very slow. And because these genes are so big, so development of therapeutic interventions became extremely difficult, unless now with the new strategies, so there are some attempts trying to use by pharmaceuticals and by scientists to be able to rescue some of these cases. Now, most of these genes are also present in several tissues of the body, which makes it a lot more difficult even to work with. Now, us genes also undergo alternative splicing, and this activities is not only a tissue specific, it's also a species specific, which makes our knowledge on this gene or these genes are still in progress. Now, because it affects several organs, now making animal models for it has also been very challenging, and that makes also the potential for us to develop therapeutic intervention is also challenging because you really need to have good animal model to apply the therapeutic design that you develop. Okay, so our interest in USH2A, which is the one that I will speak on the rest of my talk, has stemmed from the Foundation Fighting Blindness invited me to give a talk to patients with retinitis pigmentosa, and it turned out that a big portion of these patients that attended that meeting were affected by USH2A. And during the luncheon, I got together with a mother that she was devastated to tell me that her son are born with a disease called USH2A, and he cannot see and he cannot hear, and how devastated she is. And she begged me to work on it. And I said, lady, I work with peripherin and diseases associated with peripherin. And she said, please work on Usher. I said, okay, I promise you I will do that. And that was almost 15 years ago. Anyway, so we did. Another thing is that this disease is affected by congenital hearing loss and early onset blindness. And the interesting thing, beside it affects 50% of all ASH cases, it also associated with 19% of all autosomal recessive retinitis pigmentosa that are non-syndromic, meaning that only they have retinitis pigmentosa and no hearing loss. And we have managed to understand that, and that requires another talk. Anyways, the phenotype in USH2A patients are highly complex and also heterogeneous. And even with the same mutations, they found variabilities in the onset of the disease, severity, and in the progression. So that speaks for the complexity of the disease itself. So the gene is extremely large. It's about 800 kilobase and spans 72 exons in the retina and 73 exons in the cochlea. And why, that is another challenge to know. And encodes very large protein that is called Usherin, which is about 600 kilodalton. And as I indicated earlier, it's highly common among all USH cases. And this gene, which USH2A, responsible for 80% of all USH2 cases. So that tells you how common it is. So we felt like spending some time on this gene would be very interesting. Some patients with this disease have been benefited from the cochlea, but not very long. And the mutation that got us interested in the gene to begin with is the C2299G. That's the elimination of the G. And we will talk about that later. And just notice the location of this mutation within the protein. And this mutation accounts for 16 to 30% of all USH2A cases. So it's a very common mutation and located in exon 13. So Usherin, which is the protein of USH2A, has been detected to be present in 2 isoforms. That is so far 2 isoforms, but later on we'll see what happens in my talk. So we go to isoform. It's not known, that's in a human, it's not known, it's suggested to have one single isoform in the mouse. The isoform A, it's 170 kilodalton, and it's a secreted protein, which is this one. Isoform B, it's transmembrane associated protein with majority of the protein outside the cell to the extracellular space. It has very small C-terminal domain that it has the BDZ binding motif. And this is the motif that anchors this protein to the rest of other slew of proteins, which mainly USH1 proteins that anchors it to the plasma, to the cytoskeleton, which we'll go through this later. Now, the difficulty working with this protein is because it has a number of basement-like membrane domain, mainly laminin and EGFP laminin and fibronectins, which makes generating antibody for it is very difficult. So we really tried for several years with companies trying to generate antibody throughout the protein, and we failed to get a very good antibody. Okay, so it's present so far at the periciliary ridge of the photoreceptor cells right here. Okay, and present as the ankle link of the developing stereocilia of the cochlear hair cells to give us this beautiful staircase of the stereocilia. This is really exciting type of structures. I mean, you see the photoreceptor cells and the outer segment you fall in love with, but you look at the cochlea, it's really amazing organ. The exact function of Usherin is not well understood yet, but it has been thought to play an important role in the maintenance of the cell and in the development and also in the synaptic function. Okay, so our interest working with Usherin has been really to investigate the role of it in the retina and cochlea, and we included the brain, but we are at the very, very beginning steps looking at it in the brain, and I will tell you later why. And our goal is to understand the mechanism. Once we know the function of it in the normal scenarios, we wanted to see when it gets mutated, how is it affecting the structure and function of these cells? And of course, with all of that, once we learn a lot about it, our ultimate goal is to find a therapeutic intervention for it. Hopefully, that could be a long term, but we hopefully can take these studies all side by side if we can. So as I said earlier, June and others have found that the protein is located at the pericilia. Initially thought it was cilia and pericilia together, but later on it got corrected with immunogold labeling based on her awesome antibody that she used, and she kindly shared it with us. She found it is a purely periciliary located membrane protein. And at the pericilia, it associates with VLGR, which is another USH2 protein, and with whirling, as I indicated earlier, to make this complex, which is the periciliary USH complex. The idea of it is to build a collar around the cilia to help align the cilia straight, for protein transport to the outer segment, and also for the health of the cilia to hold the photoreceptor upward. So with the help from June and using her antibody that is at the C-terminus of the protein, we did immunohistochemistry, and we confirmed in our hand that it is truly located at the cilia, connecting cilia in the synaptic terminals. This has been shown by others, and we went and co-labeled the cilia with acetylated tubulin, and we could see true association between the cilia and the pericilia, close proximity. This is our old images, and now we were able to even isolate the cilia labeling from the pericilia labeling with USH2A. We looked at the pericilia, and we noticed very interesting labeling. It has a puncta labeling very similar to that we see it at the connecting cilium, but it has patches of labeling that are different. So we got interested in this. So we co-labeled it. This is a work that just sent to me about 2 days ago before I was coming over here, which is still preliminary. And we are looking at verifying a lot of its function in the pericilia. And by labeling it with PKC, which labels the bipolar cells, rod bipolar cells, we did not see colocalization, which shows interactions or a proximity, which suggests that the Usherin labeling is pure precilia type synaptic terminals of the photoreceptor cilia and has nothing to do with bipolar cells associations. When we labeled with PMCA, which labels, PMCA labels the cone and rod pedicles, rods and cone synaptic terminals, the membranes, and we could see this area where it hugs this type of patches in here. And as I said, this is very, very preliminary data, but I thought to include it here because it's interesting. So now we think these are associations between cone pedicles and bipolar cells that in this area. More labeling is coming, hopefully soon when Lars does this analysis. We'll share it with you, hopefully, in publications. So as I said earlier, that USH2A, here we have only one isoform depicted in this cartoon because the short isoform has not been thought to be present in the mouse. and it binds to VLGR through the PDZ domain that links it to Werlin, that it has the PDZ domains, and these are PDZ binding domains, okay? And as I said, that anchors it to USH1, slew of USH1 proteins, and then to the cytoskeleton. So we looked at the presence of other associated proteins to Usherin, which is VLGR and Werlin, and using the antibody that generously provided to us by UVA, we were and some also by June, we were able to see localization extremely similar to that of Usherin. If you remember at the synaptic terminal, you could see the same patches are present there. And we have not done co-localization of Usherin with VLGR at this region yet, but hopefully these type of analysis will be happening sometime soon. Werlin have also the same type of localization at the connecting cilium at the synaptic terminal. Now, we were able to look at the connecting cilium, and this work is published, I think, in the Nature Communication, is showing the connecting cilium labeling with acetylated tubulin, and VLGR is side by side to it. And the same thing, Werlin. And then we label all of these together. You know they are all located in the same area because they are partnered with each other. Okay. Now, in the cochlea, if you're not familiar with the cochlea, which is right here, it has two types of hair cells. Outer hair cells, which you have 3 rows of them, and 1 row of inner hair cells. Now, during development, Usherin and interacting partners have been responsible for making this complex, responsible for the ankle links that are needed to hold these stereocilia, which these are the stereocilia for this hair cell, to hold it together. So then the other tip links to be developed and located with the idea to get into the tip links once they are developed, to connect them so they can all be responsible to the waves of the sound. These ankle links disappear in the matured cochlea, okay? So they are now needed. So there are a transient need for Usherin and interacting partners of Usherin to be present during development of the stereocilia, then they go away. And also, the same idea, the same proteins get together to make this. So we looked at this with the help of Dominique, that he taught our graduate students. She went to his lab for 2 weeks, learned how to do whole mount stereocilia, whole mount of the cochlea. And here we are zooming at the inner hair cell and outer hair cells. And we labeled it with EPS8, which labels the tip of the stereocilia. And we labeled it with Usherin in here at the C-terminal, VLGR, and Werlin. And you could see they are located at the ankle link. You can see it a lot clear in the inner hair cell, but this is how you get it from the outer hair cells. It's always not so clear because the stereocilia in the outer hair cells are shorter, much shorter. So it's hard to get to see those in whole mount. Now, the labeling for all of these 3 basically disappears in P10. And a little bit we can still see of the Werlin to be located at the tip, as you can see it here and you can see it here. Okay, so although Usherin is very well known to affect the hearing and the vision, but there are some indications in the literature, which we knew about it over 14, 13 years ago, that it also affects the brain, affects the olfactory, the digestive system. Basically, I learned that when I met with the patients. They have hard time getting pregnant. The male is having hard time having children. So there is a reduction in sperm mortality and all of that. We observed a very similar observation in our models, which I would not be able to talk about it. this today because we are still working on it. And beside all of these organs, we have also investigated other organs that they do have Usherin, and all interacting partners are located in there, and we still don't know what they are doing there. And we do see in the disease model abnormalities in them too. We'll see where we go with that. Okay, so because we are interested in Usherin in the normal scenario, so we generated two models. One model is by, this is a knock-in model, where we knocked in the YFC with 3 flags so we can have a good handle on the protein. And in the next one, we knocked it, this is, when we knocked it is after the secretory sequence in here. Here we knocked it in after the transmembrane domain, but we have mSherry and 3 MYC tag. So that will allow us to see the proteins and track them. So the first, we looked at the wild type, as I showed you earlier, but here with the N-terminal knock-in model, we could see a localization very similar to that of the wild type, here using the flag. And here we're using the YFP with the acetylated tubulin. Here you're using H2A antibody, but with the flag. So you could see them all located. So it just looks like a normal wild type Usherin. We were very happy. And the same thing with the C-terminus knock-in model, we also see location very similar to the wild type. So because we have fluorescent protein, so this is very early trial. We use the STED to look at the labeling at the connecting cilium. And again, this is very preliminary because we did this so we can hopefully buy the STED and sometime soon come into our university. So we could see the labeling is a puncta at the connecting cilium. And when you look at the cross-section of it, you could see it as a rounding. So it's really not at the cilia, but at the peridicylia. And we hopefully in the future, we do some more creative imaging that we will be happy to collaborate. I see Chris turn his head to the other side. Yeah, we can collaborate, Chris, so don't worry. Okay, so both knock-in models are absolutely normal. The function is at 180 days, look like a normal. So we are hopefully in the future use this model for a lot of other analysis. Keep in mind, not only for the cochlea and the retina, but also for other parts of the body. Now, because we have this model, so we have this opportunity to look at the protein. I have to tell you, this is the most difficult Western blot ever. I'm a biochemist in training. This is the most difficult Western blot ever we are able to do. This is unpublished results. It shows when we used the C-terminal antibody, we can see 3 isoforms. This is regularly, we see 4 isoforms, 2 large ones. And one is 150 and one is about 120 kilodalton. And then when we use the knock-in model, we see the same isoform as well, which it tells us probably there are 4 potential isoform in the retina. And then when we used the N-terminus hair flag, we see the presence of 2 isoforms. So this 2 has the N-terminus and the C-terminus. So these other 2 probably, they have only the C-terminus, and we don't know how far they extend. But we don't know what the function of these. We are still confirming this. Unfortunately, this labeling, we have difficulty detecting it, maybe because it's the epitope is not detectable. When we do it on Western blot, we get a lot of mess. Lars is still working on it. Hopefully he get to identify bands that are associated with this. Okay, now in the cochlea, we also looked at the location of the C-terminus and the N-terminus. We both see them located at the same area, similar to what I showed you. And functional testing of the cochlea using ABR and DPOE all showed normal hearing, meaning that absolutely normal. So we are good to go with this 2 models to go and dive into several types of investigation in organs that we have never, ever dived into, but we will see how the future will take us. Now, let's go into the last model, which is the disease model. So we made a knock-in model for the C2299DLG. In the mouse is located in exon 13, in the human, located in exon 13. In the mouse is located in exon 12. It is the conversion, is the deletion of one guanidine. Deleting G leads into a glutamine conversion to serine. In the mouse, it will extend to 71 amino acid before encounters stop codon. But in the human, it extends to 20 amino acid before it encounters stop codon. So we had a long thinking decision to make. And at the end, we decided to introduce the human extension rather than the mouse, because ultimately we wanted to work toward what happens in the human. So we did the extension after the deletion of the 20 amino acid, and we put 3 flags so we can track the mutant protein and iris with GFP. GFP will allow us to know which cells expressing it. This model we generated before the other 2 models that I showed you. And this GFP had helped us to know where other tissues are expressing, where Usherin actually is expressed. So that was very helpful to us. So at the end, you get GFP and you get the truncated protein. So we generated antibody against the 20 amino acid, and you can see using this antibody, it shows the presence of the truncated protein. And using the H2A antibody or the Usherin antibody, we don't see any of the wild type. And then when you use the flag, you also see it. And of course, here's a GFP. It shows GFP only present in the photoreceptor cells. That tells us Usherin is purely present in photoreceptor cells in the retina. Okay, so Usherin is very well regulated with total messenger RNA here. We looked at it. It shows at, I think, is this the recent, maybe not, okay. Okay, yeah, it's okay. So we looked at the Usherin in here. We looked at the level of expression at postnatal day 1 and 15 and 30 and so on in 1 year, and we see a great deal of regulation of the knock-in message to that of the wild type. The higher message, we see it at P15 and then levels up in older animals. So we looked at the protein under reducing condition, and we observed 1 bound at 110 kilodalton size. And you could see the presence of GFP in here and the knock-in protein, the truncated protein. When we look at non-reducing condition, we found only a single bound when it's compared to the reducing condition. So we know that that means this protein is really not associating with anybody else. So we went ahead and looked at it in native condition. We also see it present alone, meaning that it is really not complexing with anything else. So on immunolocalization, look using the flag, and we observed localization of the mutant protein exclusively to the inner segment and nothing around the connecting cilium at all. So that has got us to be thinking about where it is in the inner segment. When we co-label with acetylated tubulin, you could see there's no localization at all, not like you could see it in here in the wild type. So we thought about, what is this protein doing in the inner segment? It's supposed to be outside because it has the secretory sequence. Then we thought, okay, let's look at it to see if it is glycosylated or not. And sure enough, we find it to be glycosylated. Now, cytosolic protein, truncated protein, and being glycosylated, and we see it in the inner segment. In another study, we colocalize it with Syntaxin, and it does colocalize with Syntaxin in the inner segment. So we are surprised as what is going on. Where is this protein? So we did cell fractionation, and by taking fresh retinal cells, put it in isotonic buffer for 20 minutes, and then isolate the soluble material outside the cell. And then we took the rest, precipitated and homogenized it, and spin it hard to isolate the cytosol from the membrane. Then when we looked at it, what we found in here, in the wild type retina, Usherin is exclusively present in the membrane, and we don't see it outside the cell. I don't know if our isolation is not good enough to isolate the protein that is strongly associated with Usherin from the outside, but we don't see it. We repeated this so many times, we're not able to see the wild type, small secreted isoform A outside the cell, but we see it inside the cell in here, which is associated with the membrane. Now, when we did this on the homozygous mutant, we don't detect Usherin in here, but we detect exclusively in the cytosol, nothing associated with the membrane. So this tells us this protein is cytosolic and glycosylated. So in what form is it inside the cell? And that's what we are trying to figure it out. What is it doing? How it can be cytosolic, but yet glycosylated. So it has to be encapsulated in vesicular structures, attempting to be secreted out, but they are not conducive to be secreted out. So we are working on, this is also helping us to think about mechanism of synthesizing Usherin and directing it to the cilia or outside the portion that goes outside the membrane. It goes to the extracellular space. Okay, so looking at the function, ERG for rods and cones, and at different time we see a reduction in the ERG signals, maximum intensity here, similar to that seen with late onset retinitis pigmentosa. We also see reduction in the cone signals, and that is also confirmed on optokinetics reflex. We do see reduction in the detection with their cone vision, and also some reduction in the number of cones when we count it. So we know that cones are also affected by this disease. So because of the proximity to the connecting cilium, we decided to look at the connecting cilium to see if it is affected. So we looked at the length of the connecting cilium in the wild type and in mutant, and sure enough, using different labeling with centrin, acetylated tubulin, we see significant reduction in the length of the cilia, but nothing, and also, but nothing in the width of the cilia, but we see significant reduction in the length of the photoreceptor. What is that? The length of the photoreceptors. Yeah, it's reduced. I'm sorry, okay, this is at different ages. I'm sorry, this is at different ages. So here's at P30 and 300. So the reduction in the length of the connecting cilium happens as early as 30 days of age and continue even in the older. Okay, so next we looked at the width of the outer segment and the width of the connecting cilium using electron microscopy images, and we do see a significant reduction in the width of the outer segment, but no effect on the width of the connecting cilium. So the absence of this complex at the periciliary area had obviously affected the structure of the connecting cilium. Okay, so because the interaction of Usherin with other proteins, in this model, we looked at these proteins as well, and what we see interestingly, that VLGR, which normally locates with Usherin, as you can see it in the wild type, and also Werlin and Usherin locates together, in the mutant, VLGR, it's not located with the acetylated tubulin at all. So mislocalization of Usherin had also mislocalized the interacting partners. And not only VLGR, also Werlin is severely mislocalized. And here it shows using the flag, which looking at the mutant protein. Here with VLGR, you can see no localization of VLGR, that the pattern we normally see it. You can see both of them are even not localizing with each other. And here Werlin also is not located with each other. So the mutant protein had mislocalized its interacting partner. Likely the absence of this complex at the pericylia had literally affected the structure of the cilia. And we'll show you later, it could affect also the trafficking of protein to the outer segment. So we next ask, I mean, would the heterozygotes have some effect? And when we looked at it in here, it's interestingly, we see some of the truncated Usherin, as you can see it here with the flag, do locate to the connecting cilium, meaning that the wild type allele had dragged some of the mutant protein to associate with the connecting cilium, as you can see it in here. And that is the endogenous allele. You could see it with the H2A, where it doesn't detect the truncated Usherin. But here's where we see the truncated Usherin, they do colocalize, as you can see it in here with the wild type. So that's exciting for patients if you, and I'll show you that even further later in the next part. In the heterozygotes in here, when we do flag and Usherin, you see they are together. That means this wild type can drag the truncated protein, and here's the flag, and here's Usherin. So that's exciting. And also exciting to know that, that both VLGR and Werlin, they do also partially localize to the connecting cilium in the presence of one wild type, Usherin. So that is very helpful to tell us that patient can benefit. Oh, I don't know if this is the last version. Okay, sorry. Oh, here you go. I went the wrong direction. I'm sorry. Okay, so here when we look at the association of acetylated tubulin with Werlin, we could see it. Some of it goes to the connecting cilium. This is in the heterozygotes. And here, VLGR also colocalized to the connecting cilium along with Werlin, and here they all together. Then we looked at the histology of it. We don't see any degeneration even at 500 days. So it corrected all the mistakes that the mutant, the homozygous, we saw in the homozygous. And the function is absolutely normal for rods and cones. So this is a good news for patients to tell them that gene augmentation or gene correction, it could be a good strategy to rescue H2A associated diseases and phenotype. So this is the premises that we are working with, hopefully, in the future. Now, next we ask what happened to this mutant protein on the cellular level in the presence of wild type alleles. So we did fractionation, and sure enough, we see portion of it. It goes to the membrane, but majority is still in the cytosol. So that portion in the cytosol doesn't have any side effect. Obviously, as long as we can get some of it to go to the membrane. And one wild type allele, and that's why it is recessive, is good enough to rescue the disease. We also see rhodopsin mislocalization to the perinuclear area in the mutant. And in a recent study done by Stephanie, she looked at the total amount of rhodopsin, and we see significant reduction in the level of rhodopsin, which is, again, as Vladimir, to our hopefully future collaboration, another point to think about it, how can this can be super active when it comes to ERG, but then rhodopsin level is reduced. That's another story we can talk about it hopefully in the future when we get it to publication. Conopsin significantly mislocalized in this model. Why? We don't know. Opsin is more so than S-opsin. We could see it at early on. Here's 200, but in 500, all catch up, where we could see it clearly mislocalized. And that's probably why we see a reduction in cone function. So we attributed, am I good with time? Yes. Okay, so we attributed the phenotype in the retina is due to mislocalization of the mutant protein to the inner segment and even eliminating it from the synaptic terminal. But we still see VLGR at the synaptic terminal, and we see the absence of VLGR unwhirling from the connecting cilium, and that affected the has partial effect on the structure of the connecting cilium and interrupted some of rhodopsin from going to the outer segment. So we next paid attention to the cochlea. As I showed you earlier, how important Usherin to the cochlea, and we looked at the total message in the cochlea. As you remember, it is important during development, and what we see, the maximum message happen at postnatal day 1. It drops to 15 in postnatal day 15 to almost like one third of the message, and then disappears in the adult animals. So we looked at the protein on Western blot and working with the cochlea. It's another difficult thing to do because it's extremely small, and you take in cochlea from postnatal day 1 and 3, it's really small. So this took Lars a long time to be able to identify and see it. So we see the same size of a mutant protein, similar to that in the retina, and fully disappear in the adult cochlea, as you could see here in P30 cochlea. And he did some immunoprecipitation from the cochlea, and he was able to see it. This is all published. We looked at the cochlea. We see GFP in the cochlea early on, and we later on went ahead and looked at the function of the cochlea using the average ABR pure tone. This is a waveform to show the earliest sound pressure that the animal could detect, usually at 10 dP in the wild type. Is the same as the heterozygotes animal, but in the homozygotes climb up to 40 dP. So we know these animals have a problem hearing. This problem hearing congenital happens as early as we were able to do the hearing test. And doing the ABR, which is here, it shows at different ages, it shows we see an abnormalities in hearing at the lower frequency at P30 and continue at the same rate up to P200, and we stop testing anymore because we know it's congenital and not progressive. So whatever happened during development, it happened and stayed and affected the hearing. But the outer ear cells, which we detected by DPOAE, it was not affected. This was a bit unusual, and that's what we had to discuss it quite a bit in the paper that we got it published in Communication Biology. It's slightly different than patients. Patients claim to be having some effect on the outer hair cells because this detection is for the outer hair cells. ABR detect both outer and inner hair cells. Then we went ahead and looked at the cochlea. We did a whole mount of the cochlea, and the cochlea is a spiral. So when we open it, we chop it into 5 pieces. We go from the base all the way to the apex here. And then we look at the outer hair cells and the inner hair cells as depicted in here. So here's a whole mount doing at all these 3 points from the wild type, and look at it from heterozygotes and from the homozygotes mutant. We did not see any differences between heterozygotes and wild type, and that's what being a recessive disease. It's very clear. But when you get closer to the homozygotes, you could see going from the base all the way to the apex, you see significant abnormality in this orientation of the stereocilia. This is simply because the stereocilia of the outer hair cells are short in nature. So obviously the ankle length to development of the ankle does not have as significant effect as you see it in the inner hair cells, where it goes from the base smaller and then gets bigger as it goes to the apex. So that's why we see the abnormalities as you go out to the apex, which is here, number 5 region, and as you can see it. So we went ahead and measured it. Ryan, who is Crane, who did this study, he looked and measured the number of hair cells that actually have misoriented stereocilia, and he found 80% of them in the apex, in the apical region have misoriented stereocilia, about 50 in the middle and 15 in the base. And he looked at the length of the stereocilia, like we saw in the photoreceptor. We thought it might be affected, but obviously it did not get affected at all in the mutant. So we looked at the outer hair cells. We did not observe any abnormalities in the outer hair cells at all regions, which coincides with our functional testing that the DPOE analysis was absolutely normal. And here we see outer hair cells normal, while the ABR was abnormal, and that coincide with the abnormal orientation of the stereocilia. So we went next to look at the cross-section. We did a lot of work on whole mount, where we're trying to identify where is the mutant protein is located. We never were able to see it. So we thought, oh my God, what happened? We can extract it, we can see it, but what happened? So we had to learn very quickly. We have to do cross-section, and that is not an easy job. Only a few labs, they do cross-section, and being amateurs in this field, so it took this graduate student a long time to learn this, but he did an amazing job. And here we labeled with phalloidian to map the structure of the of the hair cells. So here's in the wild type, you see inner hair cells, and this is like the 3 rows of outer hair cells. And you could see the red line in here. It's just so it orients you what you are looking at. So all of this labeling didn't show anything in the wild type. When you get closer into the mutant, you could see here the flag where it's truncated protein again, and the hair cells are mislocalized to the body of the hair cells, not where it's supposed to be outside in the ankle lengths. So it got mislocalized, and you can see the cells is filled with GFP, which what we expected. And here's the colocalization of the flag with, and of course, this is at P30 and P5. We can still see some P7, it gets reduced, and P10 is all gone. So the regulation even of the mutant protein is very similar to the regulation of the wild type. But the funny thing is that disappears both the GFP, which GFP here is regulated by the promoter of Usherin, which it tells us all of this regulation has happened on the promoter region and level of expression. And here is just to get you closer look at the presence of GFP, and here is the mutant protein is to the. So we next looked at, is this similar to the retina with VLGR and Werlein mislocalized? And so we co-labeled here in the wild type, as I showed you earlier, and here's the comparison. You could see at P5, Usherin is not present. This is a whole mount again. So you could see Usherin is not present. VLGR is present at the ankle link, which is very interesting. That means VLGR can get out without Usherin in the hair cells, but not in the photoreceptor cells. And it takes with it Werlein. You can see it present in here and in the tips. It's hard to see it in the outer hair cells. And then at P7, we see early departure of VLGR from the ankle lengths. You can see it compared to the wild type. And these are the outer hair cells. We could see Werlein still stays at the tip a little bit continue even at P10 at the tip, but then disappear from the rest. So this is early SEM done in collaboration with June Young, and also confirm the abnormal structure of the stereocilia at the base, at the apex of the inner hair cells, as you can see it, and not so much problems in the outer hair cells. And we could see that a lot more in this region, and not so much in the base area of the inner hair cells. So the defect in the cochlea is due to the presence of the truncated protein that weakens the development of the ankle links, the complex in the developing here. This is the wild type, but it really weakened it in the mutant, which leads into the longer stereocilia to be misoriented in a structure likely to be like that. And this had affected the capability or the significance of the inner hair cells at the base from hearing the sound properly. So in summary, I have shown you that in the retina that RP happened in USH2A is a consequence of, we believe, a structure abnormalities that happens at the photoreceptor cilia that affected the trafficking of protein to the outer segments, and mislocalization, and that's also due to mislocalization of Usherin interacting partners to be in the proper area. And we do have some other abnormal behavior that we detected in this animal in terms of vision that we are puzzled by it. That's why we approached Vladimir to see how we can collaborate with him to do single cell recording to see if this is coming exclusively from the cell itself that behaving very abnormally, or it's coming from the connections to the synaptic terminal, because the synaptic terminal also loses the Usherin in it, and there must be something happening that makes the function to be so highly abnormal. From our work, we think that gene augmentation can be a strategy to rescue USH2A disease. In the cochlea, as I indicated, that mislocalization of Usherin, it leads into abnormal structure of the stereocilia that affects inner hair cells a lot more than outer hair cells. And I'm happy to answer any questions.
So let me start with the question related to stereocilia and interlink, as you had proposed in your model that they're forming a dimer, the extracellular domain of Usherin. So that would suggest you have a protein in different directions. How that interaction?
No, yes, that was the idea before, is that you have the cilia, you have the pericilia, right?
Yeah.
And you have the first model proposed when this came about is that you have Usherin at the stereocilia, at the cilia and at the stereocilia. And you have all of these extended proteins. They do interact, they making like zipping this two membranes to each other, making the domain so highly confined to each other for transport, for interactions, for support, for all of that. But recent study showed with EM, very clear EM, and with our work, it's only present at the pericilia, and the interaction between Usherin and VLGR, we do know from work from June, it shows there are some interaction with the extended domains outside the cell, outside the membrane in the ECM. They do interact. And also UVA showed there are some interaction between these domains. But majority of the interaction is happening from the PDZ binding motif at the C terminus of both VLGR and Werlin, VLGR.
But that
And
intracellular,
Huh?
that will be
That's
intracellular.
intercellular, right?
So extracellular, how is that Usherin interacting?
I
with
don't
whom?
think we do know 100%, because majority of the study that shows there might be potential interactions is co-expression of domains in tissue
But
culture.
you have shown on your model, right?
That's model, as you, if I showed you that model, back again, do you want me to go back to it?
Oh, yeah.
Absolutely. That model is very carefully drawn not to over. Oh, that's at the beginning. Okay. I got dizzy. I'm just kidding. Oh, here you go. So you could see we did not do any connections between this and this because it has not been tested in vivo, in vivo or in vitro with the entire protein expressed in tissue culture. I mean, you have to understand the cDNA of this protein is almost 20 kilobase, which we have it in expression vector. But to make sure that you can express it in tissue culture, it's also not easy. VLGR is bigger than Usherin. So in order for anybody to do a full association outside, it's not an easy.
But that's in photoreceptors. How about in stereocilia, in hearing?
We don't know.
Right, because it is initially formed and then
Disappears, right.
So there must be some other interaction to hold that structure together. Wouldn't that
Okay,
be
so
major focus on finding how that interacts? Because
Okay,
it will give you a sense.
That is very, okay, so you could get to see this. So when this happening at early on, this to hold like the base of the stereocilia, all the proteins that are associated with the links, intermediate links and the tip links, they are synthesized and they migrate gradually. It has been shown by by whole mount analysis by other people. They show that they are migrating to their locations and develop these links with the idea to get to the tip link where cadherin and procadherin get together to build the tip links on the top here. And then kinocilia will disappear. Once that developed, the kinocilia will disappear afterward, okay? And then when this established and everybody goes to their destination, then there's no need for this.
No, I do understand that.
Because
I just don't know how that complex is formed transiently.
These complex, oh, this one?
Yes.
This is that's what happens during development. It only gets expressed and they go there and the gene gets shut down.
But do you know how that structure interacts with each other?
I don't think anybody knows.
Because that would suggest from the model that you have dimeric form of two proteins, two Usherin, reaching to each other, but that could be biochemically testable, right?
Not easy. Chris, if you wanted to, we'll be happy to collaborate with you because it's not easy at all. The cochlea is extremely small. You have no idea dissection is not easy. I mean, that's why we generated the two knock-in models that hopefully on the structure, not only on imaging, we can do such assessment on imaging. Maybe that will answer using the different ways of imaging.
Okay.
We'll see.
We have now easy question. Let's go to Professor Fliessler.
Muna, this was a spectacular presentation. My questions are much more simple than Professor have been before. You told us that there's a glycosylation, at least one glycosylation consensus site on Usherin. Is that correct?
There are more than one.
Yes. And these are N-glycosylation or O-glycosylation?
This is, we did complete glycosylation. We did not look whether N or O.
Okay, so you would be able to tell because it would be asparagine versus a serine or threonine for the attachment. But let's just say that it's N, okay? Has anybody done a point mutation to knock out the glycosylation consensus sequence, and what is the consequence of that?
Of course not. Of course not. I mean, hey, I don't think so. There's not a lot going on this protein, Steve. It's 600 kilodalton a protein. Who's gonna go in to knock in the glycosylation? Right now, no, I think the more pressing issue is what are these mutations doing on the protein itself, on the function. So no, nobody had done such a study yet. No,
Understood.
it would be
And
great.
then so the correlative question, or what prompted me to think about this, is, are there known mutations of those glycosylation consensus sequence that cause?
Not that I know of. Not. No, I don't know.
Okay.
Yeah, which is
Thank
very
you.
interesting. And I think glycosylation is a big deal in the function of this molecule, or even association, because it is heavily glycosylated. And we think that is our next step to do, really, is to deglycosylate the protein and run it on the gel and see the upper two bounds if they go into one single bound, which allows us to see, the number of isoform, pure isoform, not that they are glycosylated isoforms. You see what I'm saying?
Yep.
So that is our next step. Because also the protein is very prone to degradation because it's at a very tight area. Extraction method and all of that, biochemically, we have to make sure it doesn't get degraded, so we would not compound our results with artifact. So
I'll talk offline with you about some possible experiments.
thank you.
Okay.
Alex.
Thank you so much for this very interesting talk. Actually, I had a very similar question to Dr. Fiesler about the glycosylation. I'm here in the room.
Oh.
Right here. Yeah. Yeah.
Looking at Brian. Brian, I thought you were asking me questions.
Yeah. So
I'm sorry.
did I understand correctly that there's only one glycosylation site on this whole huge protein? Or because the shift
no.
that you observed on the gel was pretty significant. So I was actually wondering how many sites are contributing to this shift.
I really think that in silico analysis shows there are many potential glycosylation sites. And to be honest with you, I did not have the chance to go after the region of the glycosylation, as you noticed from our talk. And the reason we thought about glycosylation, because we saw the protein in the cell, inside the cell. We thought, okay, inside the cell, that's what prompted us to think, is it glycosylated? And we were shocked when we saw it glycosylated. Yeah, and that what it makes us think now, why is it glycosylated and inside the cell, and how is it encapsulated? That's our next, hopefully, attempts to do, is how is it encapsulated in vesicular structure? And that probably will help us to know how the wild type is synthesized and complex, probably with other interacting partners to go to the destination, or it goes to the destination and then associate with interacting partners. So, I mean, hopefully in the future, we'll see what the research leads us to.
Hey, Mona, lovely work, really exciting. And I love your hair cell studies as well. That's really cool.
Well, thank you.
I have two questions. First is about cones. So you showed us that at P200 and P500, there is a cone opsin mislocalization.
Yeah.
You showed a photopic ERG phenotype. But is actually Usherin expressed in the pericilium of cones, and is the cone phenotype direct, or is it the result of the rod phenotype?
Well, it is supposedly present in the cone stereocilia.
Do you see early ERG or mislocalization phenotype in cones? If you look at like 1 month or 2 months?
So we see mislocalization of M cone as early as P30.
Okay.
And then it exacerbates further in older animals. And that affects the ARG in older animals even further. And I have a slide at the end that was just sent it to me again by Lars at the synaptic terminals, which it tells us that probably the function abnormalities in cone It's not so much related to mistrafficking or mislocalization of cone opsin. Maybe it's a problem at the synaptic terminal of the cone.
Okay. And my second question is rhodopsin for the rods, is rhodopsin mislocalization the main phenotype, or what other roles could Usherin play in rod photoreceptors other than regulating rhodopsin targeting to the outer segment?
So level of rhodopsin, which I didn't present it, but that's the work of Stephanie. We are now doing different type of investigations, trying to hopefully develop therapeutic intervention for it, especially for rhodopsin, because it plays a huge role in retinitis pigmentosa, we think, in this model, and probably in patients. So when she looked at a careful analysis of rhodopsin level, we observed significant reduction in rhodopsin level, full extraction of rhodopsin. So that means that is beside portion of rhodopsin mislocalized to the pericilia. And we don't know why is it mislocalized to the pericilia. We didn't see mislocalization of peripherin or other outer segment proteins. We did not see mislocalization of CNG, for example, to the plasma membrane, but we don't know. Yeah, we're still working. I mean, this is a new model, we're still working on it.
Very exciting.
Thank
Hi,
you.
Dr. Naash.
Hi.
Beautiful talk. I loved your immunohistochemistry.
Thank you.
It is my understanding that Usherin is involved in intercellular trafficking too. And given the localization that you were showing today, I was wondering if you think that Usherin also plays a role in intercellular vesicle trafficking. Given that it's in the,
Why would you think that?
because the localization that you're showing,
Well,
like in the ribbon synapse, in the periciliar zone, and that's, formation of vesicles there.
It plays role in taking, it's all suggestive. There are no direct experiments to indicate it is exactly what's happening. It's thought to help trafficking of proteins to the outer segments. But to be honest with you, that was at the beginning when people thought it is present at the connecting cilium plasma membrane, but it is not there. It's not present at the connecting cilium. But because it's pericilia, so a lot of proteins insert themselves to the pericilia and they go up to the outer segments for morphogenesis, right, of the outer segment. So because that complex is messed up at the pericilia right now, so therefore trafficking of some protein has been delayed. I don't think it is significant a trafficking problem in here. And the reason we see problem with opsin, because there's so much opsin going to the outer segment. And you have here shorter cilia and misalignment of the pericilia to the cilia, rhodopsin, it's going to be delayed. I think it's a secondary effect. I don't really think it's really the main effect. I feel like when it comes to rhodopsin and opsin, it's secondary effect. And I really think that the main defect we are seeing here, I really think is still working on it, but the main effect is the absence of the protein at the synaptic terminal, the absence of the protein at the connecting cilium had messed up the ability of the photoreceptor cells to do proper and regulated function. And that function could be vision. And because of misregulating vision, we don't see, the cell cannot handle it over a long period of time. That's why we see very, very late onset phenotype. Does that make sense, Vladimir? Yeah.
Very interesting talk, Dr. Naash. I had a question. Did you ever try to look the ciliary changes in patient mutant cell line of ASH2A?
Me?
Yeah, did you try looking the ciliary defects in patient-derived fibrous cells?
No. I am at the bench. I never I never touch patients at all. Yeah, I'm at the bench. I let the doctors do that, the ophthalmologists.
Hello, Muna.
Hey,
My question is somewhat related to Vladimir's, but about transducing. Do you see in your truncation mutant model, do you see any change in a total amount of transducing and especially localization in
I thought you never going to ask. Nobody's going to ask me this
in,
question.
in synaptic
Yes,
in synaptic terminus? Do you see
okay.
any changes position there?
So the very first time we sent the paper out, which was like almost 6 years ago, we sent it to PNAS, and it was completely structured differently. It was a lot toward function, a lot toward mislocalization of transducin and restin. So then it's sent back to us to say, no, this is not what we were hoping to see. That's not, we like it, but we're not gonna accept it. So then we went back and we did, that's when Lars joined our group, and we completely restructured the paper, and that's what we have in Nature Communication. But we do have a great data to show that arrestin is significantly slowed down in translocation to the outer segment in light-dependent manner. Transducin is also translocation of transducin, slightly delayed, but more so arrestin than a transducer. Again, we are still trying to figure it out. Why is this? And that's what hoping that Vladimir, a new collaboration through Vladimir, that we hopefully can understand the biology of what's going on in this photoreceptor cells. We are very, very excited and interested in sharing these resources with anybody who wants to do the collaboration to investigate as what's going on, because, you know, we are too old to stay in this field. Okay.
Yeah, good talk.
Thank you.
Yeah, I have a question about your model. And so your model, you can cause the deficit in the vision and the hearing, and it's really good. But my question is on the ERG. On your ERG, you see the deficit about, even at the 500 days, and it's almost old age. And you see the deficit of about 10 to 20%.
Which about 35% in the older end, yes.
But
we see
in the human, the vision can become blind, right? And do you think this is match for the human vision, for the model?
Okay, that's very good question. I'm sure you have read our paper, because that's exactly what was one of the reviewers' comments, is that how close is this model to the patients? Yes, we do see some differences between our models and the patients, and also to the knockout models. Although our model has the truncated protein, but basically it mimics the knockout, because the protein is there, but it's not making it where it's supposed to go. The knockout doesn't have the protein at all. And, but I think that the differences between the human and the mouse is with the structure of the retina probably attributed to this minor differences. But if you remember at the beginning of my talk, I made it, you know, I stated that this gene and USH2A cases and the phenotype, even within the same mutation, within the same family, it is so heterogeneous. And the heterogeneity comes from the other allele. I mean, if you have a mutation, and there are several, I mean, it's diving into this gene and what happening in patients is very, very interesting. In some scenario that they have compound mutation, where they have one allele is C29G, while the other allele is another mutation. When they combine it together, the phenotype is completely different, both for the rods and the cone, and the cones, and for the patient, for the hearing, and let's not mentioning other organs. So the heterogeneity and the environment, since our animals are absolutely homogeneous, coming from the same line, the same background, the same strain, I think this isolate the effect of the mutation absolutely a lot more pure than we can compare it to the patients. And I think that might have some differences in there. I mean, it's surprising to see the outer hair cells absolutely normal, okay? But the inner hair cells is messed up. So nobody wants to ask me about other organs? Okay.
I do.
Oh, here you go. That's why.
Oh, that's excellent.
Yeah.
Because, well, you focus on those, so maybe not exactly about other organ, but other cilia. These are very specialized cilia. They are very static, but there is a lot of very dynamic cilia, including even in RPE upon stress, like starvation. So I wonder if Usherin is expressed there and maybe temporarily just upon the stress, or whether it has a role.
You mean in the RPE?
Like in the primary cilium, you know, those small ciliums that appear after in starvation conditions. So, you know, also stem cells have the cilia. Is it there? Is it?
I have no idea. The only thing I can say, if you're talking about the retina, other cells in the retina, it is 100% rods and cones. Because
but you have wild type conditions. You didn't starve these animals.
I
for
didn't.
example.
Well, I'm just speaking out of our observation, because GFP, we see it only in the photoreceptor cell, and GFP is controlled by the same promoter. So, and it disappears at the same time. So that tells me that Usherin is purely expressed under normal condition in rods and cones. But we never stress the animal. We never put it in high intensity light, I mean, we are so busy in what I have showed you that I have to put our effort where it's supposed to be, you know? So no, we did not stress the animal to see other ciliated protein. It might be turned on. But I think not every ciliated cell in the body express Usherin. I don't think so.
So which one do? Because you have
Okay,
seen
which
it.
one do? Okay, I tell you, we see it in the brain. And when our student showed GFP localization in the brain, and don't ask me which cells of the brain, because we are very preliminary in that one, we looked at the behavior. And we did the maze, and we did the social behavior, and that's part of her thesis. And it's absolutely, I mean, you can even genotype the animals from their jumpiness. The GG mice, which we call them GG mice, extremely jumpy. I mean, it takes really a special person. Jumpy, they very disturbed. But we did social analysis. They all failed. They don't like to socialize. They don't like, or they don't go where they're supposed to go, because really their hearing is affected. We see the sperms mortality is incredible. I mean, they don't survive. They do die shortly after isolation. Their number to begin with, I mean, we have very tough time getting the male to produce. The female don't bear the children. I mean, we get 2, 3 offspring to survive. So we looked at the ovary. The ovary, we could see GFP in the ovary, and we have not done this analysis in the new ones yet. The wild type knock-in, we haven't done it. I mean, it all needs a lot of hands to do it. So the lab are more focused right now on the important experiments, but we are hopefully to do all of these analysis. But we see accumulation of white material in the ovary that we have no idea what they are in the aged ovary. Then what else? In the olfactory, we see it. You know, these are all organs. I mean, we have to learn how to dissect and all of that. So, I mean, if anybody wants to collaborate, we'll be happy to work together and figure it out more as we can in the near future.
We had a great conversation, so let's finish with the final question.
All right, thank you.
Thank you so much, Dr. Munad. So all your figures were self-explanatory. They were amazing.
Thank
So
you.
I have a couple of questions. So one mutation-related question and one phenotype-related question.
Okay.
So did you have a chance to look at, so for example, now we are predominantly looking at a deletion 2299. So did you look whether this deletion affected any signal sequences like exonic splicing enhancers causing aberrant splicing?
No.
Okay, because
No. Well, speaking of that, you're absolutely correct because alternative splicing is huge in Usher gene. And we did not want to spend effort on that. The reason is that work came from Netherlands, where they did make the animal models for intron 40, pseudo intron 40. I was so interested in making mouse model for that and designed it and ready to let it go with the cost to make this pseudo exon. I thought it was so cool. But they did it earlier and they published it and it made me think, okay, I don't want to waste our time. It turned out it didn't work in the mouse. So no, because it is a mouse, that might be completely different alternative splicing than in the human. We decided not to go. I mean, isolating the RNA and getting effective cDNA library from it is not, for us genes, is not trivial, yeah.
Because I was interested, because I worked on a nonsense mutation that affected a ciliary gene, and then we predicted that the mutation has affected a potential exonic splicing enhancer. Yeah.
Oh, we could do that. That's easy to do by
Yes,
you know, just amplifications across that area.
Exactly.
Yeah.
Yes.
Yeah. Well, it depends when Lars has the time to do them. It's in the to-do list because one of the things to look after isoform is to amplify by doing a QRT-PCR. And amplify specific regions and to see in the sequence which exons are present and stuff like that. And this is like to-do list.
Okay. And one last question.
Yeah.
So which phenotypes occurs first? Is it the vision loss or is it the hearing loss? And
which one happens?
So which phenotype occurs first? Is it going to be the
hearing loss? Yes.
Okay.
Hearing loss and disturbance, which I think related to the brain, activity of the brain. We're not so knowledge in the brain, but we've been so busy even to go across the street to Baylor and tag along with a brain person. But that's our interest in the future.
So do all the patients have issues with all the organs?
Yes. I noticed that 15 years ago in the patients. They were so disturbed and unbelievable. And they go and complain. Their personality is different. I mean, these people cannot see you, cannot hear you. They are not aware of their surroundings. The doctor says, you know, loss of hearing, loss of vision is what makes these people very uncomfortable. It's not that. It's their ability to analyze things. And I think I noticed that from the beginning. And that's why I made sure that when I designed the the disease model is to have GFP in there so I can see where is GFP, because it's really difficult to have faithful antibodies for all of these large Usher proteins. Because of the basement membrane. So it was always misunderstanding. Where is it located? What is it doing?
Thank you so much.
All right, let's join Brian Jones to thank Muna for her presentation.
They see me, right? Thank you so much, guys. Thank you all for staying to the last minute. This is fun.